Okay, so welcome back to this next video in which we are discussing the transduction of painful stimuli. Okay, right, so now what we want to turn our attention to is how protons, a too high concentration of protons within the extracellular fluid, can also activate the axon terminals of nociceptors. Okay, right, so I'm going to give you another reference now for the information that's going to be within this video. And my next reference is another review, this time by Kellenberger and Grutter, okay, and it was written in 2014. Okay, so this is Kellenberger and Grutter. Okay, and basically they have written a nice review on the ion channels that are going to be involved in the transduction of nociceptive proton concentrations. Okay, so this is Kellenberger and Grutter, and the review was written in 2014, so it's a nice up-to-date review. Okay, right, so basically the ion channels that we are now going to discuss are what are known as ASICs, okay? And what this stands for is acid sensing ion channel. So the A is for acid, the S is for sensing, and the I is for ion, and then the C for channel, or since I've put an S on the end, channels. Okay, so we want to discuss these acid-sensing ion channels, which basically are going to be ligand-gated ion channels, which are activated by protons binding to their extracellular domain. Okay, so let's have a look at the structure of these acid-sensing ion channels. Okay, so basically they have a very similar structure to the P2X receptors. So the entire ion channel is made up of a trimer of 3 separate subunits all joined together to make the entire thing. So here is the ion channel, here's the pore through which the ions will move, and then we've got 3 separate subunits all joined together to make the full ion channel. Okay, like so. Now, the same protocol then as for the P2X receptors, what we'll do is we'll take out a single one of these subunits of the acid-sensing ion channel and have a look at its membrane-spanning topology. Now, once again, the top will be the extracellular fluid and the bottom will be the cytoplasm. Okay, so you have the amino terminus of these proteins which produce these single subunits of an acid-sensing ion channel within the cytoplasm. Then they have a membrane-spanning alpha helix. Then they have an extracellular loop, and this time it doesn't have the fancy structure that we saw in the previous video for the P2X receptors, okay, of the 5 cis loops. And then they have another membrane-spanning alpha helix, and then their carboxylic acid terminus is within the cytoplasm. So a very similar membrane-spanning topology actually to the P2X receptors, and in fact the acid-sensing ion channels are grouped with the P2X receptors in a certain family of ligand-gated ion channels known as the P2X-like receptor family of ligand-gated ion channels. Okay, so ligand-gated ion channels are categorized into 3 categories. Okay, so one family is known as the P2X like receptors, okay, and this is the family which the P2X receptors belong to and also these acid-sensing ion channels belong to. Then there is the glutamate-like receptors, which contains the AMPA receptors, the NMDA receptors, the kainate receptors, which are all ligand-gated ion channels. And then finally there's also a family of ligand-gated ion channels known as the nicotine-like receptors, which has an old name as well, which is probably the name that's more often used for this family of ligand-gated ion channels, which is the cis-loop ligand-gated ion channels, so named because it absolutely, definitely is known that they have a cis-loop within their extracellular domain. Okay, so these are also called the cis-loop ligand-gated ion channels, and this family is by far the most studied family of ligand-gated ion channels. It contains the nicotinic acetylcholine receptor family of receptors. It contains the GABA-A receptors, the glycine receptors, the 5-HT3 receptors, etc. But the important thing for us is that there is this categorization of the ligand-gated ion channels into these three separate families. And one of these families is this P2X-like receptor family. And both the P2X receptors and also these acid-sensing ion channel receptors, they are both within this P2X-like receptor family. Okay, right. So, again, the simple thing would be if there was just one subunit of an acid-sensing ion channel, which was called the acid-sensing ion channel subunit, and all you did was you made three acid-sensing ion channel subunits, join them together to make a trimer like so, and that would be very, very simple. Okay, it's not that simple in reality. In reality, there are many different genes for acid-sensing ion channel subunits, and more complicated than that, there are different splice variants of each one of the genes. And if you don't know what that means, don't worry, I'll explain. Okay, so basically, there are 4 genes which code for acid-sensing ion channel subunits. Okay, so there is the ASIC1 gene. Okay, then there is the ASIC2 gene, then the ASIC3 gene, and then finally there's also the ASIC4 gene. Okay, so there are these 4 separate genes which all produce subunits of acid-sensing ion channels. Okay, now there is actually 8 known subunits of acid-sensing ion channels. So at first this looks like a bit of an anomaly until you remember the principle of splice variants. So let me show you the principle of different splice variants that come from the same gene. Okay, so let's just remind ourselves of the central dogma of biology. Okay, so let's take, for instance, the ASIC1 gene. Okay, so let's say this little strip of DNA here, and this is what this is supposed to represent, not the plasma membrane anymore. This is supposed to represent now a double-stranded piece of DNA. So we've got one strand and then the complementary strand here. Okay, now let's say that this is the gene for ASIC1. Okay, so the ASIC1 gene here. Now basically, there is only one of the DNA strands that is actually going to be used by RNA polymerase to make the mRNA for that gene, okay? And that strand is known as the coding strand of the gene. So the strand that I'm now colouring in turquoise, this can represent the coding strand of the ASIC1 gene. Okay, so this is the strand which RNA polymerase 2 will actually work its way along and produce a piece of mRNA which is complementary to. Okay, so here is the piece of mRNA that's being produced that is complementary to that coding strand. So this piece of mRNA will actually have the same sequence of organic bases as the non-coding strand of DNA, the strand that was complementary to the coding strand, because both the non-coding strand and the mRNA are both complementary to the coding strand of DNA. Okay, of course you're going to have to take account of the fact that this is mRNA and this is DNA, so we've had the thymine organic bases in the DNA replaced by uracil within the RNA. Okay, so basically this mRNA that we produce that is directly complementary to that coding strand of DNA is not actually ready yet to be translated into a protein. So you cannot just stick this piece of mRNA through a ribosome yet. Instead, it is known as pre-mRNA. Okay? And the reason that you can't just stick it through a ribosome at the moment is that there are loads of bits of this that are not actually supposed to be translated. Okay? So, there are portions which are supposed to be translated, and these are the portions which I'm now going to box, basically. And these are the portions called exons. Okay? So these are the portions that are actually supposed to be translated. So these are called exons. Whilst the portions in between the exons are sometimes referred to as the junk portions, and these are called introns. Okay, these are not supposed to actually be translated. If you translate these, you'll get some sort of junk, basically. You'll get a terrible sequence of amino acids, and you'll get a non-functional protein that'll probably aggregate together and form plaques, okay? So not good. Right, so what you need to do, basically, is process this pre-mRNA. You need to chop out the introns and then re-stitch the exons back together. And this process is known as splicing. And it will finally produce a much shorter piece of mRNA known as the mature mRNA, which is basically the exons stuck back together. So this is the mature mRNA. Okay, right. Now, sometimes there are more than one different pieces of mature mRNA that you can produce from the same piece of pre-mRNA. Now, how does this work? Well, let me give you a simple example. Sometimes there are exons which are optional, okay? So little small exons where you can include them, or you could just chop them out, basically. And if you include them, you'll get a slightly longer protein when you finally actually translate the mature mRNA. And if you don't include them, it won't harm you. You'll still get a functional protein. So basically, you can produce multiple different mature mRNAs from the same pre-mRNA. And these different mature mRNAs will then go on to produce different proteins. And these different proteins are then the different splice variants that you get from the same gene. So basically, a single gene can produce multiple proteins, and these multiple proteins come about by variations in the splicing process and are hence called the different splice variants of that gene. Okay, so each of these 4 genes for acid-sensing ion channel subunits have multiple splice variants. So for instance, ASIC1 has ASIC1A and ASIC1B. Okay, which are the 2 separate splice variants that come from the same gene. ASIC2 has 2 separate splice variants called ASIC2A and ASIC2B. ASIC3 has 3 different splice variants known as ASIC3A, ASIC3B, and ASIC3C, and ASIC4 has only 1 known mature piece of mRNA that can be produced, and therefore only 1 known protein. So that gives us a total of 8 different subunits. 1, 2, 3, 4, 5, 6, 7, 8. Okay, right. So, now what we can do is we can combine these different subunits together to make trimers, and then it's the same problem as we had with the P2X receptors. We can make homotrimers, where you combine 3 identical subunits together to make a trimer which is made up of 3 of the same subunits, and that will be called an ASIC homotrimer of whichever splice variant you produce. Okay, so whichever particular of one of these 8 subunits you use. You can also, however, make heterotrimers, in which you mix and match, basically. So you use multiple different types of ASIC subunits when you're actually producing your trimer. Okay, so there are homotrimers and heterotrimers. So now what we need to discuss is how are these actually going to activate nociceptive neurons. Okay, so basically in nociceptors, which are sensitive to high proton concentration in the extracellular fluid, okay, you will have these ASIC channels. Okay, so certain ones of these ASIC channels will be found within your nociceptor neuron. Okay, and basically, when the proton concentration is too high within the extracellular fluid, i.e. when you have an extracellular fluid acidosis, okay, which is a too low pH. So pH should usually be around 7.5. You find that these ASICs become active at around a pH of 6.5, although it does vary depending on the exact subunit composition of your acid-sensing ion channel trimer. Okay, so what happens is when proton concentration gets too high, i.e. the pH becomes too acidic, protons will start to bind to your acid-sensing ion channel and they'll cause it to open. Now when it opens, it's permeable mainly to sodium ions. It is permeable to potassium as well, but it's actually around 10 times more permeable to sodium than it is to potassium. Okay, its conductance to calcium is very low, so we don't need to worry about that. So it's going to allow sodium ions to move through it and also potassium ions a little bit, but we know of course that the electrical potential difference across the cell membrane is usually going to start off at around negative 65 millivolts, so potassium movement out isn't going to be that large anyway, so we might as well just focus on the movement of sodium in. So as soon as you open these channels, you are going to get a net movement of positive charge into the cell in the form of sodium ions, and this will cause a depolarization of the electrical potential difference across this cell membrane. That's called the receptor potential. Some of this sodium will diffuse into the action potential origin down here, cause a depolarization of the electrical potential difference down here, and then that will cause the action potential origin to fire an action potential. Okay, so that now concludes our discussion on the transduction of painful stimuli. We have looked at the nociceptors, we've looked at their anatomy and their physiology. We then had an in-detail study of the different nocicensors which you have on the axon terminals of the nociceptors and how their respective noxious stimuli can actually activate them.